Hi, welcome to my walkthrough of the Genebass browser. Please see the description to find a link to the browser, the mini project contributors, and a link to the medRxiv preprint that was submitted describing important analysis details and methods used in the study. To get started, I'm going to show you a page displaying association results for a single gene phenotype pair. There's a link to this page in the description if you'd like to follow along. This is the gene LDLR and the phenotype disorders of lipoprotein metabolism and other lipidemias. On the left-hand side, we have details about the phenotype, including identifiers, descriptions, category information, and information about how the phenotype was determined, as well as important analytical details like the number of cases and controls. There's also a link to the official UK Biobank showcase website if you need more information about the phenotype. The goal of Genebass is to evaluate the impact of rare coding variation that we've measured through exome sequencing, particularly the effects of predicted disruption of protein coding genes on human phenotypes. We display the results of gene-based tests alongside detailed data about the individual variants included in those tests. Using this control panel, I'll simplify things by unchecking all columns. For each row in this table, we have a variant ID comprised of chromosome, position, reference, and alt allele delimited by dashes. We can see the RSID by clicking this checkbox. I'll also click the consequence column and the HGVSP column to see the predicted impact of each variant on the protein product. In our study, we considered 3 functional annotation categories for variants. In red are predicted LOF variants or PLOF variants. In yellow are missense variants, which also include low-confidence PLOF variants and in-frame indels. And in gray, we have synonymous variants. Damaging substitutions from the first 2 categories may result in haploinsufficiency and hence disease, or in some cases have a protective effect. The goal is to test whether we see an increased allele count of cases relative to controls, so I'll enable those columns and also the allele frequency. You can look at the case and control tracks and see the presence or absence of each variant in cases or controls, represented visually along the exons of the gene. The legend over here shows how the circles are scaled by allele frequency. We use the mixed model framework SAGE-Gene, which includes single variant tests, so I'll check the p-value and beta columns to show the values from that analysis. Single variant p-values are plotted above on a double log axis. We can see that these variants are rare and don't have strong associations. In rare variant analysis, it's useful to use the protein coding boundaries to increase analysis power for detecting associations at the gene level. We ran 3 group tests for each annotation category: gene-based burden mean tests, scat variance-based tests, and scat-O, which uses a combination of burden and variance component tests. This gives us a 3x3 matrix of annotation categories across the tests. We also ran extensive quality control on the summary statistics, cumulative allele frequency filters on the genes, and genomic control or lambda GC for each phenotype and each gene. Pay attention to the green indicators to ensure that you're only looking at the highest quality data. You can hover over top of each indicator to see the acceptable ranges.